All right, so today we're going to talk about summary statistics. These are simple measurements of central tendency in a data set, measurements of spread. We're going to talk about types of variables, and then I'm going to give you a little bit of an introduction to probability. Actually, I'm going to do all those things in slightly different order. I think we start off with probability. So here we go. Why statistics? First of all, what do statistics do for us? They just exist to torment us. Some people would think that. A lot of people do not appreciate the study of statistics. It's one of those things when I tell people, I teach statistics, they think, ooh, this is not someone I should be friends with. I actually love teaching statistics. And we really need to understand statistics as scientists. They're really helpful, especially when you're dealing with the messy world. And for ecologists, the world is often a very messy place. That doesn't mean we're the only scientists that need statistics. All scientists need statistics, but they're particularly helpful when you're dealing with really complex systems. So we're going to start off just with an introduction to probability. We need to understand probability because it helps us understand how likely certain events are. And this statistical analysis is basically trying to understand the likelihood of particular outcomes. So what are the patterns that we see, and how probable are those patterns due just to chance alone? That's a really good question. How probable are any patterns that we find? Are they very common? Are they very rare? And how likely is it that the pattern that we see is due to just random chance? So nature is full of pattern and is full of noise. So the goal of most natural sciences are to find the pattern in the noise of nature. And statistics is a really great tool for that. So we can increase our chances of seeing a pattern through the fog when we increase our sample sizes and when we design really clear experiments to try to test particular research questions. So once we've collected data, we need statistics to interpret them, to show us the patterns, to help us see if it's all just random noise or if there's actually a pattern that's interesting. And I think this is why I think statistics is really fun, because humans are pattern seekers. We're always looking for patterns. And so it's really fun. It's like digging for buried treasure, I think, when I run statistical analyses. And the other thing that's really great about statistics is that it's a common language across all scientific disciplines. So all disciplines, whether they're natural sciences or social sciences, rely on the same basic types of statistics. So probability and uncertainty. Probability is everywhere. So if you start looking into it, you'll find that the weather forecast gives you a measurement of how probable different types of weather phenomena are for the coming day. So the forecast is based on years and years of past data, and the probability of rain in the future is based on how often we had conditions like we have, coming up, and how often those conditions resulted in rain. So when you see there's a 40% chance of rain, that's a probability statement. Similarly, if you're doing some hydrology work and the river starts to rise and you get to what's called a 100-year flood stage, that's also a probability statement. It's a statement that the river would only get so high once every 100 years. Another good example of probability would be on the back of any lottery ticket. They'll give you a chance of winning. Maybe you have a chance in one out of every 3.45. That's a probability. It's a pretty high probability. Chance of 1 in a million would be a much lower probability. So just a little side note, since we're dealing with data and we're dealing with different data sets, that you can have differences in both precision and accuracy in your data. And so, for example, precision is defined as agreement among values. So you can have an instrument giving you very precise numbers, like it gives you 1.12, 1.13, 1.11, right? So all the values are really close to one another, but that instrument might not be very accurate. So the 1.13 that it's telling you might be really far off if it's measuring something like nitrate concentration in your water and your actual nitrate concentrations are like 2 or 4. Then you have an instrument that's very precise. It's good, it has good agreement, but it's not very accurate. So accuracy then is how close is the value to the truth. So if you measure something on, a mass balance and it's a very accurate mass balance, it means it's been calibrated and it's giving you a result that's very, very close to the true value. So those are just, very fine points, but really important points when we're talking about data and analysis of data. So for an example around probability, you can think about a pitcher plant like the Sarracenia purpurea, and it's a kind of plant that extracts nutrients from insects that fall into its pitcher and drown and then are dissolved by these kind of digestive juices. The plant itself, in terms of capture, is not terribly efficient, so a lot of insects escape. They're not actually captured. And so we could measure the probability of successful capture events. And so, for example, an event in this sense, this is just some definitions, an event would be an insect visits a pitcher plant. That would be a single event or a trial. And then the outcome of that event could result in 2 ways. One could be prey capture, and the other could be prey escape. And so the outcome of each event could have multiple options. And then replicates of those events would be sitting and watching many insects visit the plant and then recording the outcome. So you say, oh, here comes a bug. Oh, it escaped. Oh, here comes another bug. Oh, it escaped. Here's another bug. Oh, that one got captured, right? So that would be the data set that you'd be collecting. And then your experiment would be that set of trials that you decided, I'm going to sit here and watch this plant for 10 minutes, or I'm going to watch this plant for 10 minutes and then another plant for 10 minutes, or I'm going to watch 100 plants and I'm going to watch each one for 10 minutes over the course of 10 months, right? So different experiments can be designed in different ways. And of course, the more events that you have, the closer you might come to the true probability. So for example, the probability of a particular outcome would be the number of outcomes divided by the number of trials. So for example, the probability of prey capture would be how many times an insect was captured divided by the number of trials, the number of insect visits, basically. And if the probability is 0, then that event never happens. If the probability is 1, then that event always happens. And for example, if the probability is 0.5, the event would happen about half of the time on average. So can we measure all the pitcher plants in a population? No, we can't. We can't have a single person sitting at every single pitcher plant in every population of pitcher plants around the world, right? So no, we can't measure all the pitcher plants in a population. But we can estimate the probability of prey captures by pitcher plants by taking samples from a population and by summarizing the data from all of the samples in something like a histogram. And a histogram is a graph where the y-axis is frequency and the x-axis is outcome. And so say we spent a week and we spent 1 day watching out of every week, sorry, every week we spend 1 day watching 1,000 insect visits. And for each week, then we determine the number of prey captured and we plot the data using a histogram. And maybe we get something like this. So the number of observations would be the number of times a certain number of prey were captured. So for the y-axis here, we have the number of observations, and then how many times was just 1 insect captured? Well, twice. How many times were 2 insects captured? 6 times. How many times were 9 insects captured? Looks like 5 times. So pause the video and figure out what's the most probable outcome from this histogram. Okay, great. Hopefully what you found was the most probable outcome is the one that happens the most often. So it's most probable that about 7 insects are captured. You can see there's a lot of variability here, so you might not feel very confident about that number 7, right? And that's part of statistics, is that feeling confident in your result. So we can't measure every pitcher plant in a population. We can estimate the probability of particular outcomes by taking samples from a population. And then if we do this many, many times, we can calculate a mean expected value, or what's called an expectation. So that's kind of an introduction to probability. I'm going to briefly describe the different types of variables that you're going to come in contact with in this program, and kind of the general types of variables that exist out there. The first are discrete variables. This is any variable that you can measure using an integer, like a count. So like how many individuals are in a plot, what is the age, right? You tend to give your kind of year-based age, like I'm 14 or I'm 19, not 19 years, 11 months, and 2 days or something like that. So that would be something discrete. Continuous variable is any variable that measures along a smooth number interval. So temperature, that is something we often measure, like 26.3 degrees Celsius, for example. And then categorical variables are variables whose values are not numbers, but they might be words like colors or success or failure, capture or escape, yes or no, presence, absence. Those would all be categorical variables, as would things like the species of tree that you're looking at, or the location on the landscape. Are you in a lake or a pond? Are you in Olympia? Are you in Wisconsin? Something like that. Okay, so in the realm of statistics, I'm going to use my hands here. It might get a little bit strange. So we're interested in these big populations. Okay, so population at the population level, this is what we're really interested in. If I have a question about oak trees, I want it to be about oaks, right? Not just like one oak, but a population of oaks. And so we're really inherently interested in what are called population level parameters, but we can't know anything about that. We can't measure the whole population almost ever. And so if we do try to measure a whole population, it's called a census. And we're in the middle of the 2020 census right now. But, like censuses happen in the US once every 10 years. They're really hard. They take a lot of time and a lot of energy. And so for the most part, we don't do census work in ecology or in other disciplines, but instead we estimate population parameters by taking samples from populations and calculating what are called statistics. So a parameter is like, say, the mean mandible length for a specific ant in the whole population. That would be the parameter. You might measure a bunch of ants. You might sample a bunch of ants from a population and measure their mandibles and calculate the mean value from your sample. That would be a statistic. And so this is, I'll come back to this a lot, this difference between populations and samples, between parameters and statistics. But populations have parameters and samples have statistics. And I hope that becomes clear throughout the course of this program.